All right. Yes. So good morning, everyone. So as we know that we are trying to do the revision of histopathology, then we will go to the actual subject, which is histological techniques and immunohistochemistry. I'm telling again, this subject is immunohistochemistry. This is not histopathology, but immunohistochemistry and histopathology are closely related. So that's why let us brush up this histopathology. Then let us correlate how immunohistochemistry is different from histopathology. Okay. It will be great if you mute yourself, students. Okay. Please mute yourself. If you have any doubts, you can unmute. If not, it's taking too much time for me to track you, to mute you. Anyway, so yes. So in a classical histopathology, what we will do, we will take the biopsy or autopsy. Biopsy is from a living patient and autopsy is from a dead body. Upon taking a piece of tissue, we will convert the semi-solid or gelatin-like tissue to solid tissue. Then this solid tissue will be cut into slices with a specialized instrument called microtome. Upon getting the slices, we will stain them and we will observe them under the microscope. So finally, we will observe the tissue under microscope upon staining. And that is the primary agenda of histopathology. And the primary application of histopathology is to identify cancer cells. So cancers can be identified in their earlier stages, and the chance of treating the patient is high upon detecting the cancer in the earlier stage. And that can be facilitated with the help of histopathology. Okay, so as we already seen these things, let me go to the next slide. So the subject is immunohistochemistry, as I stated. So let me directly go to these things. 1 second. See, we got a biopsy and we will do some processing steps. Upon processing, we can able to observe the sections under the microscope. So this is a summary of histopathology. But okay, if we are taking from a living patient, it's called biopsy. If you are taking from a dead body, then it is called autopsy. If you are taking biopsies from a fetus, then that can be stated as a fetal autopsy. So the idea of fetal autopsy is, here we are trying to figure out what is the reason for the fetal death. Upon immediate, after birth, if the fetus is died, then we will go for investigating the cause of the fetal death. So by this, we can able to predict the future possibilities of miscarriage by the mother, and we can able to counsel them if there is any genetic problems in the history, or the reason of the fetal death can be okay, it can be investigated, it can be thoroughly rectified, and we can able to take some preventive measures for the mother in her next carriage. Okay, so that is why we will also do this fetal autopsy. Okay, so let us see the major steps in tissue processing. As we already know, the first and foremost step will be collection of a sample, which can be a biopsy or autopsy. Upon collecting it, immediately we need to fix the sample in order to prevent the sample from decomposing. That can be done by placing the sample in a formalin solution that causes cross-link formations in the proteins, so that the tissue won't be damaged by any microorganisms. So upon fixation, we will go for sample sending. Okay, sample reception, sample identification, and labeling. So usually this this will be done in the surgical ward. Then they will send this sample on a jar, in a jar, and they will send to the lab. Upon receiving the sample, this is also a very important area where we need to observe very keenly. Okay, upon receiving the sample, the first thing that we need to do is we need to cross check the details on the sample with the requisition form, whether the requisition form details are matching with the sample labeling or not, whatever there in the jar. And also we need to thoroughly inspect physically the content inside the jar. Say for example, in the requisition form, they said a biopsy of tongue, and in the jar we got a eye. Okay, they said tongue, but there is eye in the container. Shall we go for processing this tissue? No, we should not. We should thoroughly inspect all the things, and if we have any troubles in identifying the sample, then immediately we need to call the corresponding surgeon or the doctor who sent this sample. And also we need to ensure that there should be patient age, name, age, sex, referred to physician, physician address, location, time of sample collection, everything need to be there. If any data is missing, at any cost, we should not process these samples. Unidentified, incompleted, improper samples should not be processed. If you feel any sympathy or kindness, ayo, the patient is dying, I need to do the test, then you will be in the legal proceedings, because it is something, a very delicate process. They're taking sample from surgery after surgery. So patient is not ready to give to undergo surgeries. Okay. So unidentified and improper labeled samples never process. Okay. We should not process such samples. Okay. Then, okay, everything tallied. Okay, the requisition form and the data and the labeling and the content in the jar has been tallied. Then the second thing we will do, the next thing we will do is cross examination. So here we will physically inspect the organ. So in cross examination, can anyone tell me, students, what parameters we will observe in cross examination? Do you have any idea what parameters we will observe in cross examination? Since you are masters, I hope that you are very good at these things. Can you tell me what we will observe in cross examination?
So the appearance, the
Appearance, color,
the quantity,
quantity,
and the quality of the sample.
quality of the sample. Very good. I need at least 10 to 15 points. Now tell me. I need 15 points. Next, any more? Cross examination is very important. Most of the students I have encountered, they will say only these 4 or 3 or 4 points, and they will say that is it in cross examination. It is much more than that. It's not a simple thing, cross examining a sample. Okay, let me tell you the cross examining things. So immediately after receiving the sample, first we need to check the status of the sample. Is that organ properly fixed or not? Is that organ has putrefied? Is that organ has caseous necrosis? Caseous necrosis means the organ will become like a cheese. Okay, first we need to identify the status of the organ. It is myxoid, it is caseous necrosis, it is putrefied, it is a semi-decayed. You need to write the status of the organ. Okay, then you should write the volume of the organ, the weight of the organ, the borders of the organ, the dimensions of the organ. Dimensions include length, breadth, thickness, and volume, the overall volume of the organ. Then we also need to mention the associated structures came along with the organ. Say, for example, we received a sample like we got a uterus sample. Okay, so the patient got some uterine some problem, so the doctor removed a part of uterus and they sent to the lab. Now, along with the uterus, what structures we have, what structures included in the sample? Does it has a fallopian tube? Does it has fimbriae? Does it has suspensory ligament? Does it has a ovary? If it is ovary, we need to also identify the positioning of the organ. Is it the right ovary? Is it the left ovary? So we need to identify the orientation of the organ also. Now, if you got any problems in identifying the orientation of the organ, immediately call the doctor. Sir, is it a right ovary or left ovary you sent? Okay. Okay. So we need to also record the associated organs that came along with the specimen. Okay. After these things, the next important thing is now with the help of a sharp blade, we need to cut the sample. Upon cutting, we need to give the description of the cut surface. What is the status of the cut surface? Okay, so I cutted the organ. Upon cutting the organ, if I encountered any calcium nodules, then I need to write some calcium deposits has been identified upon cutting the sample. If you cut it very easily, if you identify any fibers, then you need to tell the it is fibrous organ. The cut surface has fibroid. If it has a caseous necrosis, sometimes upon cutting, it will be like cheese inside. And that means some part of the tissue has been decomposed, either due to bacterial infections or anything. So we must give the description of cut surface also. Then last and final, but very important, is you need to take a photograph of the sample. Why do we need to take photo of the gross sample? Because see, we received left ovary, but the patient has cancer of right ovary, but we didn't receive right ovary. Okay, now we call the doctor and we will say, sir, we only got left ovary, we didn't get right ovary. it is the doctor's mistake. He didn't perform surgery properly. Okay. Now what the doctor will tell? He will blame us. He will say that you have lost the sample and you are blaming me. You are the technician. You did the wrong thing. So that is why we must have proof of what we have, okay, about the sample. So we must take a photograph and we can show this photograph as a proof to the doctor. So this is the status of the organ. This is the parts that I received. So no one can question us. Okay. Moreover, performing the test, we need to be, so you need to mute yourself, students. Let me mute you all, okay? So we must always take a photograph of the gross sample. Only upon taking photograph, we must proceed for the next tissue processing steps. So now you understood, right, why the gross examination is important? Is everything clear, students? By the way, can you able to sync with me, students? Is there any issues with my language, my vocabulary, my pronunciations? Is everything fine? Am I going too fast? Any issues with me? Yes, students, are you listening? Are you listening, me, students? I think you're unable to unmute.
Yes, sir.
Yes, okay. Okay, let me continue. Yes, students. Next, we have fixation. So fixation, we already know. Here we will denature the proteins. We will make the crosslinks among the proteins. Then we will go for the so the tissue become hard. Okay. Upon this, we will go for the next step. Now we will remove water from the tissue, which is called dehydration, by suspending the tissue in graded alcoholic solutions. Now we need to remove the alcohol because alcohol can cause bleaching effect to the tissue, and it can also form some free radicals that may damage the tissue. So this alcohol will be replaced by a clearing agent such as xylene. And also we need to ensure that the xylene should have refractive index, which is equal to the refractive index of the glass slide. So the tissue, the tissue will be clear. We can able to clearly identify under the microscope without any blurry images. So for this purpose, we will go for this clearing step that is done with the help of this this xylene. Okay. All right. 1 second. Let me mute my phone. So that is a clearing. Then after clearing, now we will replace this xylene with other molten wax. So that molten wax is paraffin wax. So we will heat the paraffin wax and we will suspend the tissue in the paraffin wax so that the liquid wax will enter into the tissue and the xylene will be removed. Okay, and we will leave the tissue outside for next 1 to 2 hours. So the tissue will become hard. So the wax within the tissue will become hard and the tissue will become so solidified and it will become hard. Then we will place this tissue in a Luca-Tel mold and we will make a block. Okay, just like a brick, a block of brick. We will make a block of tissue. So the tissue will be in the center and it will be surrounded by a block of wax. And now this block will be placed in a microtome. Microtome means it's a specialized, it's a specialized potato grader. You know, right? We will make potato chips. Similarly, it is a modified potato chip maker. So here we, instead of potato chips, we will get human tissue chips. Okay, very thin chips in one way. Upon getting this things, this tissue sections, now we will go for staining. The usual stain we will use is H&D staining. Upon H&D staining, we can also mount it. Mounting means very simple, laminating the slide. Okay, so just like the way we will laminate our certificates or our photos with this plastic wrapper so that they won't get damaged by any environmental factors. Similarly, we will mount the glass slide with this DPX and we will go for, so the tissue will be preserved for a long time, almost 200 to 300 years. Okay, upon mounting, we will go for microscopic examination. So these are the classical steps in histopathology. Okay, now ultimately we will get this microanatomy. We can able to see the cells or the cells in the tissue in the microscope, right? So these are the overall steps in the tissue processing. So whatever I said, the same story is here. Okay, so grossing, all the things. Okay, this you already know. So I'm just fast forwarding it. Fixation, dehydration, clearing, infiltration, block making. This is microtome and we got it. Okay. So this is a gross examination, then fixation, then dehydrations, clearings, embedding and block making, section cuttings. There are certain tricks that we need to follow. Okay, well, the section should not be curled. So there are many things that we need to mind. Okay, during section cutting. Ultimately, we will get this H&D stain. After H&D staining, we can able to see the cells clearly under the microscope. But the problem with the problem with classical tissue processing is here we are damaging the tissue. We are we are damaging the proteins in the tissue. In the first step of tissue processing, in the fixation, we used formalin, and formalin literally coagulated the proteins. If proteins are coagulated, fine, the morphological structure didn't alter, but we are missing some key features in the tissue which is not visible to the naked eye. Only just for surface knowledge of cells and their arrangement and their architecture, the classical histopathology will help us. In this tissue, the tissue that you are seeing right now, this patient has a normal tissue. His thyroid tissue looks perfectly fine, but he has a serious cancer in his body. But that cancer is right in front of us and it is hiding in front of us. I'm telling again, there is a cancer in this particular photo, but we are unable to identify that cancer because the cells mimicking like they are healthy and they're fine. They are, they're hypnotizing us like the patient is perfectly fine. Okay. Now I will tell what type of cancer this patient has. This patient has multiple sclerosis. Okay, plasma cell cancer. So what is this multiple sclerosis? So multiple sclerosis means here the patient has a blood cancer. We know that the antibodies are produced by B cells in the body. So the activated B cells are called as plasma cells. So in this particular patient, there will be cancer to the plasma cells. So this the population of plasma cells has drastically increased from 10 cells to the 1 lakh cells. Okay, from 100 to 1 lakh cells, they got increased. So now you might ask me a question, sir. We have plasma cells. If we have more plasma cells, then more antibodies will be produced and we will be superhumans. We won't get any infections because more plasma cells, more antibodies, more protection, more resistant to infections. So that is theoretically what we will feel, but in reality, that will be a cancer. So this excessive plasma cells will produce excessive amount of abnormal antibodies, and these abnormal antibodies will start depositing in the tissues. These excessive antibodies will start depositing in the tissues, and these antibodies will alter the normal physiology of the tissue. So this protein deposit in multiple sclerosis, this protein is called amyloid protein, amyloid deposits. So upon this amyloid deposits, the tissue will become hard in the body. If you have amyloid deposit on the tongue will become hard. If you have amyloid in the thyroid gland will become hard. And this amyloid protein, we cannot observe in normal histopathology technique. And you know the reason. The reason is it's a protein, and this protein has been destroyed in the process of tissue processing. So we can't identify this amyloid, and that is a major backdrop of this particular histopathology. We can't able to locate proteins in the tissue. Here, for example, let us take another thing. So we know that cells have receptors, all receptors antigens, and I cannot target any antigen in this particular tissue. The reason is the antigens, all antigens are proteins. Proteins are already destroyed. If that is the case, how can I able to know in what part of this tissue my desired antigen is located? So that is not at all possible by histopathology. Then, is there any other way to detect or to localize or to pinpoint the desired antigen in this particular tissue. That means still I want tissue section, but without damaging the antigens. I still want the section, but without protein destruction. Okay, somehow I did some magic and I preserved the proteins. Okay, the antigens are preserved. Now, how can I able to locate? How can I able to track? How can I able to identify the protein, the antigens present in this tissue? And there we have these beautiful tools, and that is our antibodies. We know that antibodies are specific in their targeting. So we will, if I want to target CXCR5 receptor, CXCR5 receptor is very important receptor which is present on the surface of helper T cells. That is very essential for the HIV virus to replicate. So, so now I need to check the CXCR5 protein expression on the cells. So now what I will do, I will develop a anti-CXCR5 antibody, and I will allow this antibody to bind on this antigen. And how I will identify this antibody? This antibody will be labeled with any chromosome that can able to give color to me. Okay, so now I'm gonna use antibodies, and I'm using a specialized fixation technique that won't damage the cells, tissues, so that I can able to identify or visualize specific proteins that I want in the tissue. And that technique and that field is called as immunohistochemistry, and that is our subject. Okay, it's a modified histopathology. Here we are not going to destroy the proteins. We are not destroying the proteins, and we are trying to locate these proteins by using some specialized antibodies. Okay, hence this is called immunohistochemistry, and this is our subject actually. So immunohistochemistry has broad applications in medicine. Now you can able to identify any type of antigen products in the tissue. We have many disorders that are occurred due to protein depositions, abnormal protein depositions in the tissues, and all those disorders, you name few like Alzheimer's, neurological disorders, muscle cancers, even heart is very resistant to cancer, but still all types of cancers can be easily identified with the help of this immunohistochemistry. Okay, sometimes there will be fat deposits, sometimes there will be even enzymatic deposits in the tissues. Even those enzymatic deposits can be able to track with the help of immunohistochemistry. And that is how immunohistochemistry is different from classical histopathology. I hope this is clear for everyone. Now you understood, right, students? What is the difference between histopathology and immunohistochemistry? Any doubts? Any confusions so far? Can I proceed? Are you following me, students? Do you understand what is immunohistochemistry and histopathology?
Yes, sir.
So no doubts, right, so far? So now I will show you the definition of immunohistochemistry. Okay. Meanwhile, let us check your syllabus now. So now you understood we are going to study histopathology or immunohistochemistry. Okay. So let us see what is there in the syllabus. So whatever points I said, they are segregated into different units. Okay, that's it. You also have molecular pathology. That is a small unit in this, I will tell you. So let us see the syllabus, students. So in the unit 1, we have special techniques for histochemical studies. Here we have advanced special trainings and enzyme histochemistry. So the chapter 1 has a freeze drying and vapor fixation. So here what we are doing is we will suspend the tissue in cryo temperatures. Okay, you know, right? Liquid nitrogen. If we keep tissue in the liquid nitrogen, immediately the tissue will become block. It will become like a ice cube. It will become a tissue ice cube because the water will be freezed and it become ice cube. So here we bypassed all the steps in tissue processing. We haven't done fixation, we haven't done dehydration, that clearing, infiltration, everything has been bypassed. And also there is no damage to the there is no damage to this proteins. Okay, so that is a very nice statistical approach of dry freezing the tissue. Okay, making the tissue solid. So by this we can able to detect almost all types of things in the body. Okay, like axons, Purkinje cells, astrocytes, all fat deposit cells can be easily identified with the help of this immunohistochemistry approaches. Okay, because usually fat will be we know, right? Fat will be directly dissolved in alcohols. Fat will dissolve in alcohol. Since we are using dehydrating agent, all fats will be dissolved. So that's why we are bypassing all those issues with the help of immunohistochemistry. Then immunohistochemistry has a subdivision called enzyme histochemistry. Here we will try to detect certain enzymes present in the organs. Say for example, let us take prostate cancer. In prostate cancer, we know that prostate specific antigen, and in the prostate we have this alkaline phosphatases. So excessive amount of alkaline phosphatase enzyme in the tissue indicates the possibility of prostate cancer. So those enzyme markers can also be detected with the help of this immunohistochemistry. Enzymes such as dehydroxynases, oxygenases, esterases, tyrosinases, phosphatases, all these enzymes can be able to be detected by immunohistochemistry. As the saying goes, all proteins, all enzymes are proteins, but not all proteins are enzymes. So since all enzymes are proteins, we can able to track the enzymal activity in the immunohistochemistry. Okay, then there are other things like demonstration of various fixatives used in histochemistry. Okay, immunohistochemistry. So these the fixatives that we are using in immunohistochemistry, they are reversible fixatives. When I say reversible fixatives, yes, they will also cause crosslinks among the proteins, but we can able to remove the crosslinks. Okay, reversible fixatives. So we will use some modified fixatives in the immunohistochemistry, and we will also use modified microtomes such as cryomicrotome in the histochemistry. Okay, so this is all about unit 1. So unit 1 giving us a very basics in immunohistochemistry. Then let us go to unit 2. So in the unit 2, yes, they are going with the actual antibody detections. So here we are using fluorescein fluorescein antibodies. Okay, fluorescein immunoassays. So with the help of fluorescein immunochemistry or FIC, we will do. So here we will take an primary antibody, secondary antibody, and this secondary antibody will be conjugated with a protein with a substance that will glow under the UV light. So we will use a modified microscope and a modified antibodies that can be used for detection of specific antigens in the tissue. So we will use this fluorescein techniques for detection of all the antigens in the tissue. Okay, so now there are some technical challenges, students. So yes, I will tell in a nutshell, then we can go in depth. So some technical challenges we will face in immunohistochemistry are antibodies has two properties: affinity, avidity. Can you tell me the difference between affinity and avidity? Do you have any idea, students? What is antibody affinity and what is antibody avidity? Do you have any idea? Okay, I will tell you. Affinity means it is the level of attraction between antigen and antibody. So for example, I will give you very simple idea, students. Okay, imagine a magnet. Okay, if we have a very large magnet, it can able to attract iron pieces even from large distances. Even if I keep iron here, this large magnet can able to attract it. So the attraction of antibody towards the strength of the attraction of the antibody towards the antigen is called affinity, very simply. So the higher the affinity, the higher the possibility of attaching to the antigen. This is called affinity. And second one is avidity. Avidity means it upon attachment, how strong is the bond? Is it too strong or is it delicate bond? Avidity will tell about the strength of the bond, whereas the affinity will tell about the level of attraction towards the antigen. So attraction is affinity and bonding is avidity. Okay, now say for example, we want to detect, we are going to detect an antigen, prostate specific antigen, which is very closely related with peroxidases. Peroxidase is present in prostate gland. Okay, peroxidase also present in red blood cells in some organs of our body. Okay, peroxidase is a common enzyme in those organs. Okay, example is prostate gland. We got prostate gland and my target is prostate specific antigen. I need to detect the prostate specific antigen in the prostate gland, but this prostate gland also has peroxidase. The problem is in the steps of my fluorescein microscopy, fluorescein assay, I'm using a chromogen called peroxidase to develop the color. So one of my reagent is also peroxidase. Okay. Now the problem is my reagent peroxidase, not my reagent peroxidase, the tissue peroxidase will act. It will react with the antibody and it will give false positive results. There is no PSA antigen, there is no prostate specific antigen, but there is peroxidase. Upon adding my antibody, it will develop color because peroxidase is already present in the tissue and it will give false results to us. We will think like, yes, my targeted antigen is present. Actually, the color developed not because of antigen, but it is due to the peroxidase, which is already present in the tissue. So that is why in such scenarios, we need to mask those peroxidases. The tissue peroxidases need to be masked. So that is called blocking. How to block endogenous tissue enzymes to ensure not to cross-react with our testing protocols. So blocking of endogenous enzymes is called blocking. Okay. So for that purpose, we will suspend the tissue for a brief period of time in human serums or some other monoclonal antibodies solutions to block the endogenous activities, blocking some substrate activities. They can be peroxidases, they can be some antigens also. So there is this step called blocking. And other thing that we need to ensure is, I said right in the previously, fixation. It is a reversible fixation. So now the proteins are masked. These proteins are not detectable because they are fixed. Now we need to unfix the proteins. And this is called unmasking. How to unmask the coagulated proteins. That step also we will include in this particular thing. So those two are very important. Okay. So that is all about your syllabus. So here you can see blocking of endogenous enzyme and background and methods of freezing, controls. Then we have these things. Okay, demonstration of various types of antibodies. We have heterogeneous polyclonal antibodies, heterogeneous antibodies. So their types can be seen here. And in the unit 3 is damn interesting. It is a fish. Okay, fluorescent in situ hybridization techniques. So fish is really interesting. Here we have all the molecular things like this euploidy, aneuploidy, chromosomal disorders, all these things. Okay, so the third unit discussing about detection of genetics, chromosomes with the help of same antibodies. So we are using antibodies not at the level of tissue, but now we are going up to the level of chromosomes at the level of cells. So that is your molecular pathology. Molecular pathology is a very interesting thing. And yes, in the 10th experiment, they kept this beautiful technique called flow cytometer. So there is an advanced flow cytometer called a fluorescein activator cell sorting machine, which can able to detect almost 20,000 cells per second. And each cell, it can able to detect almost 20 to 25 parameters. That means 25 antigens in a single cell, it will detect for almost 20,000 cells within a second. So that is an advanced technique. Okay, an instrument called FACS. and that facts we will discuss in this particular subject. So these are the things that we are going to learn in the coming classes, in the future classes in immunohistochemistry. So this is all about the immunohistochemistry. Okay. So first unit, we will discuss about the basics in immunohistochemistry. Second unit, we will discuss about the procedures and the technical challenges that we will face during the procedure of immunohistochemistry. And in the third unit, we are discussing about the advanced level of immunohistochemistry, which is the molecular pathology, starting with fish, okay, fluorescent in situ hybrid.